right, so here in 18.5, we're talking about different types of immunity, and then we'll be talking about different types of vaccines and how they work for immunity. So we have two different classifications of adaptive immunity. We have our first, which is active immunity, and then the second one, which is passive immunity. And we have spoken a little bit about these already, but we're going to get into details now. So active immunity is where we actually activate the individual's own adaptive immune defenses. So what this means is the actual person that we're speaking about has to have actively fought off some certain type of pathogen in order to have our immune defenses built up against that particular pathogen. So an activation of the individual's own adaptive immune defenses. So we've gone through the process with the T cells, with the B cells, even with our innate nonspecific immune response. But here we're talking about, of course, the adaptive immunity. So this can be acquired naturally. So if you are exposed to something, so if you're exposed to the influenza virus and then you get that in your body and then your body's defenses go to work on that, your B cells, your T cells go in and they fight that off whether you get sick or not, then you are going to acquire that immunity naturally because then you will have built up those memory cells, both the memory T cells, memory B cells, and then you're going to be able to fight that off much more quickly and to a higher degree than previously when you hadn't been exposed to it. Or this can be acquired artificially. And so when we acquire active immunity artificially is when we are exposed to something artificially, and then our immune system is going to be activated because of that. And this is when we're talking about vaccines, because with a vaccine, we are artificially exposed to something, but it actually does activate our own adaptive immune defenses. So we are fighting something off, even though we weren't naturally exposed to it. So we weren't naturally exposed to it in our environment, and we went in, got it put into our bodies, and then we fought it off. Then we can divide that further into our natural active immunity. So if we naturally actively fight off something or artificially actively fight something off. So vaccines, or if we are exposed to something in the environment. Now the other side to that is passive immunity. When we have passive immunity, this is when we have the transfer of adaptive immune defenses from another individual or another animal. So in this case, what we're going to be talking about is an antidote, for example. So if there's some particular type of toxin, say a snake bite venom is in the body and it's going through the body and you go into the hospital or a clinician, and then they give you antivenom, for example, to try to stop the damage of that toxin. This would be passive immunity. Your body did not create that antivenom or that antitoxin, but what it was done was it was given to you artificially. So it came from somewhere else. So likely an animal in that case, because they produce lots of them, purify it, put it into containers, and then can pass it along for that use specifically. Or this can be done, so naturally or artificially, same with our active immunity here. Now, the one that I was just talking about with the antidote would be artificially. So it's artificially passive. So you go into the hospital and you're given the antidote, the antitoxin, and then you have been artificially given something. It's not something from the environment, and that is not something your body's fighting off on its own. Or it can be acquired naturally. So naturally would be what we've been speaking about previously as far as passing on antibodies from the mother to the child. So that's an example of passive immunity that is naturally acquired. So that child, so if the child is nursing, let's say, or still in the womb, and we're passing along those antibodies, that child itself is not fighting off the infection. So if the mother is exposed to, say, the influenza virus or something else, and then the mother is producing those antibodies to fight that off, then the mother is working on fighting something off. It's activated the mother's adaptive immune defenses, but not the child's adaptive immune defenses. But because the child is nursing, for example, they're passing along those antibodies to the child, and so that is a natural way to become passively immune. So then that child, if they do get exposed to that particular pathogen, they will already have something in their body, specifically they would have these antibodies that can then help to fight that off right away. So then we can also say we have natural passive immunity and artificial passive immunity, which is what we were just talking about. So we have these 4 different classifications of adaptive immunity, to natural, to artificial, to active, to passive. So now we're going to look at that in a little bit more detail. So I kind of already did talk about this, but so here's our natural active immunity. So remember, if it's active immunity, we have actively fought this off. So this is our adaptive immunity that develops after natural exposure to a pathogen. So, for example, if you have lifelong immunity that develops after the recovery from chickenpox. So you were exposed to the varicella zoster virus, you got chickenpox, you go through the illness, you go through the recovery, and now you have a lifelong immunity to chickenpox. So you had your immune system activated, and then it has gone through that process, and now we have those memory B cells, memory T cells, and now you can fight that off. The length of time one is immune, however, depends on the pathogen and the antigens that are involved. So you can have situations where when you're exposed to something, you have lifelong immunity, but sometimes when you're exposed to something, you only have immunity for a couple of years before those antibodies or those B cells or T cells are recycled because we haven't been exposed to it, so the body kind of gets rid of them and makes space for other B cells and T cells. So some examples here are activation by protein spikes during intracellular viral infection confers lifelong immunity. So if we're talking about something like the varicella zoster virus, this is a virus, and these protein spikes, if the antigen, in this case we're saying that the antigen are these protein spikes, and then we had this intracellular viral infection, now our body has produced these B cells and T cells, memory B cells and T cells that are going to confer lifelong immunity. However, activation by a carbohydrate capsule antigen during extracellular bacterial infection might provide a shorter term immunity. So this is similar to the biological molecules that we spoke about before, where if we have proteins, they're going to be a stronger pathogen or a stronger antigen for us to fight off, or easier for us to recognize. We see that more easily, and we have this bigger immune system response. That also means that we have longer immunity to something like that, something that like a protein spike compared to our carbohydrate capsule or DNA or a lipid or something. So we have our natural active immunity. And this is, of course, how we get most of our immunities. It's just by being out in the world and exposing ourselves to things, and then our body fights them off, and then we keep these memory cells around for later so that when we have another exposure, then we're able to fight it off more easily. Then we have our natural passive immunity. So this is our natural passage of antibodies from the mother to the child before and after birth. So again, we have our IgG antibodies. Remember that those are the only antibody that cross the placenta from the mother's blood to the fetal blood supply. So this is very important because, again, the mother is actively fighting something off, but the fetus that's growing is passively getting that immunity. Now, this immunity is going to last up to 6 months after birth. So any antibodies, any of these IgG antibodies that the mother produces in response to a pathogen are going to be going into the fetus through the blood supply, passing across that placental barrier to the fetus, and then that will last up to 6 months after birth. So that's a good protection for that newborn for the first 6 months of life. And then we have the IgA antibodies that are transferred from mother to child via breast milk. And again, that's why it's important, if one can, to breastfeed, because then you're passing on anything that you may be exposed to or anything that you have previously been exposed to that child, or the person that would be that has birthed the child has already passed along the IgG antibodies. And then if somebody is continuing to nurse the child afterwards, then they have those IgA antibodies that can be either from an active infection that's the mother's fighting off, or from previous ones that are still throughout the body. And then this immune defense lasts through the period of breastfeeding, so that those antibodies will constantly be being put into the breast milk. So as long as a person is nursing the child, then they're going to have a more bolstered immune system. So they'll have this natural passive immunity from the person that is nursing the child. Then we have our artificial passive immunity. So this is the transfer of antibodies produced by a donor, so whether it's a human or another animal, to another individual. So in this case, this is to prevent disease after someone's been exposed to a particular pathogen, or as a strategy for treating an active infection. So if somebody has been exposed to a pathogen, they might go in and get something in order to make sure that they don't get sick because of the pathogen. Or if somebody is actively infected with something and they're not doing a very good job of fighting it off, then they might give them this artificial passive immunity. So for example, if it's prophylactic, so this would be a strategy to try to prevent somebody from having an active infection. Some prophylactic examples are rabies. Right, so if somebody gets attacked by a dog, oftentimes, or a raccoon, then you should go to the hospital or the doctor's office, whichever is best or easier for you. And then when the person does that, then they will likely give them some artificial passive immunity, right? So give them a shot to try to make it so that they don't get rabies. You may not know if that animal had rabies or not, but it's prophylactic. So it's just to prevent this disease from happening. So this would be artificial because it's given separately. It's not from the natural body. And it's passive. It's not the body itself fighting something off. It's given passively from somebody else that fought something off. Same thing with hepatitis A and B. So if somebody's been exposed to hepatitis A and B, then you can have a prophylactic there to try to prevent hepatitis from causing infection. When it's used as a treatment, we use this in example for Ebola virus infections in 1995. So people that were infected received blood transfusions from people that were recovering. So those that had already mostly fought off the Ebola virus and were recovering, then that meant that they had, all of those B cells and T cells and antibodies that are floating around to fight off the Ebola virus. So they would take their blood and then they would put their blood into and transfuse it. So take the other person's blood out and put their blood in order to make sure that they were getting those B cells and T cells and antibodies to hopefully help them fight off the Ebola virus rather than getting so ill that they actually die. And so it did change the mortality rate. So when somebody was able to get this treatment for the Ebola virus, the mortality went down to 12.5%, versus with just the untreated Ebola virus is about 80% mortality rate. So a huge, huge difference there. Another example are diseases caused by bacterial toxins. So things like tetanus, people go in and get a tetanus shot to try to make sure that they don't have an issue with tetanus. Same thing with botulism and diphtheria. So these are some other examples of just artificial passive immunity. So going in and getting something that was made via some other human or some other animal to help your immune system to fight something off, or to make sure that your immune system doesn't have to fight something off. And then lastly, we have artificial active immunity. So in artificial active immunity, this is our activation, right? Because it's active immunity. We're actually activating our adaptive immunity, but this is through deliberate exposure, so artificial exposure. So artificial exposure, deliberate exposure of an individual to a weakened or inactivated pathogen, or preparations consisting of key pathogen antigens. So this is the basis for vaccines, and we're going to talk about this in more detail. So again, artificial because we're not naturally exposed to these things, and active because we actually are engaging our adaptive immunity in order to try to fight off the vaccine that was just given. So this is a table from the text that kind of summarizes these things. So we have our natural going this way, our artificial this way, passive, and then active. So these are the 4 classifications of adaptive immunity that we just went through. All right, so before we move into speaking about vaccines more specifically, we're going to take a look at herd immunity, which is kind of one of the principles or backgrounds to vaccines. And herd immunity is when most individuals in a population are able to mount an effective immune response. So whether that's naturally immune because they've been exposed to it, or artificially immune because of vaccines, it doesn't matter. It's just that they are immune to it, and that those that are unable to mount an effective immune response are then protected. So in this case, what we're saying is that if we take a look at a particular population and we say that most of the people are immune to something, then the pathogen whatever pathogen it is that these people are immune to, or most of these people are immune to, the pathogen will be unable to spread effectively. So if somebody that has the pathogen comes into this population, they may be interacting with a whole bunch of people, but the people that they're interacting with are already immune to that. So when that pathogen then gets into the person's body, that body is able to effectively mount an immune system response and get rid of that pathogen. And if they are, then those people that second person are exposed to, then essentially the idea is that they wouldn't be passing that pathogen along as easily, because their immune system would be getting rid of it so quickly and have such a quick immune response to it that then that pathogen is not going to get very far in that community or in that population. So then susceptible individuals are unlikely to be exposed to the pathogen. So those that might not be able to have immunity to something. So people that are unable to get vaccines, for example, maybe somebody that is immunocompromised can't get a vaccine because just fighting off the vaccine perhaps would kill them or bring their immune system so low that they wouldn't be able to fight off anything else and it's too big of a risk. So with the idea of herd immunity, then these people that are susceptible are unlikely to be exposed to the pathogen, because then likely if it does enter this population and it's passed along to people that are already immune, they would clear the infection, hopefully, before they would expose somebody that is susceptible. Also, then the text mentions that as new individuals enter the population, either via birth or relocation, and leave via death or relocation, that vaccines can be used to maintain a high percentage of immune individuals. So the idea is that when people are talking about vaccines, like for example, the CDC or the WHO are talking about vaccines and trying to get everybody to get a particular vaccine, they're saying that herd immunity is important. They're saying that if we get the majority of the population to get the vaccine, then these people will all be immune, and then that pathogen doesn't get a foothold. It's unable to spread effectively. That said, recall that a person is able to transmit a pathogen at any stage of infection, which means that even if person number one here is the person that brings that pathogen along, right? So they're the ones that have this particular pathogen, and they, let's say, cough it out to person number two. And then let's say person number two gets this pathogen in their body, but they had the vaccine, right? They had the vaccine. That doesn't mean that they don't have the pathogen in their body. That doesn't mean that they don't have the pathogen multiplying in their body, getting inside of their cells and multiplying happily, and getting into more cells and multiplying happily. What this does mean, however, is that that person's immune system has a response in place already, because it's already been exposed to that. So it's able to fight that off more quickly. Now, this doesn't mean, however, that this person hasn't been walking around, this person number two, can be walking around for, say, 2 weeks or more, coughing out this pathogen, just in a regular cough, or when they wipe their mouth or something with their hand, or take something out of their mouth and their saliva's on their hand or something, just everyday occurrences. It doesn't necessarily have to be cough, because of course when we think of cough, maybe we're thinking like, oh, then they're sick. But just regular everyday interactions with different people, touching different things and hugging other people and things like that. This person number two may not feel sick at all. Remember that when we have a second exposure to something, but we have already had that first exposure, we might not feel sick at all, because our body's taking care of that pathogen very well. Now, that doesn't mean that the pathogen's not in the body, and that doesn't mean that we're not transferring that pathogen. So what that means is that even if somebody is immune, even if all of these people over here are all immune to it, they can still all be exposed to the pathogen and be passing along that pathogen to anybody else. So yes, it is less likely that people are going to be able to spread that pathogen as far, just because the body will be able to clear it more quickly than a person that is fully actively infected. And, maybe it's an illness that lasts 3 weeks, and during those 3 weeks, maybe they're passing on a higher amount of that pathogen in their cough or holding the door handle or something. However, that doesn't mean that herd immunity does protect everybody, because they can still pass that pathogen along to anybody else at any point in the process because they do have the pathogen in their body. So that can get to every single person in the population that is immune could have the pathogen in their body because they're passing it along, passing along, passing along, passing along, and then they can pass it along to somebody that doesn't have that immunity. So susceptible individuals are less likely to be exposed because the body's passing it and fighting it off so quickly, but that isn't necessarily the case. So susceptible individuals may still get the pathogen, and it still may pass along to them just as easily as it would have before, because again, it can be in the person's body for 3 weeks even though they're not feeling well, and that's just a made-up amount of time, 3 weeks. But of course that depends on the pathogen and the health of the individual, etc. But they can pass that along at any point in time. So there is a lot of research being done on herd immunity and vaccines and their effectiveness. So the idea is that it won't be able to spread as effectively because people are then exposed a second time. But remember, during infection, it can be passed along at any stage of that process. So now that we're moving into talking about vaccines, we want to talk first about variolation. Now, variolation is kind of the start to vaccination. And variolation is the deliberate inoculation of individuals with infectious material from scabs or pustules of smallpox victims. So variolation in general is a deliberate inoculation of individuals with some sort of infectious material. So it kind of sounds like vaccines, right? This is where vaccination got the idea from, or those that develop vaccines got the idea from variolation. And so this was in 10th century China. What they used to do is people that got smallpox is very dangerous. It ends up killing a lot of the population, especially back then when they didn't have, sanitation and doctors and, all of these things that did a really good job and really understood things to the degree that we do now. So what they would do is they would take victims of smallpox, so those that were sick and they had scabs from the rash and pustules and things that were forming from the smallpox, and they would take material from a person that was sick, and then they would inject it into the skin, or they would put it in a device and blow it into the person's nose. So this is called variolation, where they're deliberately infecting somebody else with infectious material from these scabs and pustules from somebody that's sick. What they noted, and the reason they did this, was because most of the infections that they were passing along then were milder than the naturally acquired smallpox. So they noticed that people that just naturally got smallpox because they were exposed to it got really harsh smallpox, were very, very sick, and had difficult time recovering from the illness, if they recovered at all. But then they noticed that if they purposely infected people with stuff from the other person, that they were able to recover. It was more of a mild infection, and then their recovery time from this mild infection made them protected. So they were more protected from being around people that had smallpox. So they were able to provide this milder infection of smallpox. The person would recover, and then they could go work with people that were sick with smallpox and help them to get better, and they did not get sick again. However, some infections were more serious and or fatal. So this wasn't an exact science, obviously. Blowing material from scabs and pustules into people's noses isn't going to give us an exact quantity. And so some of these infections did end up being even more serious than if they had naturally acquired them and killed people because they were passing along smallpox, and that's very dangerous. So there were some epidemics as a result of variolation. So they were passing this along so that people wouldn't get the full-blown smallpox and perhaps die, but then this ended up being an epidemic. And then those that, similar to what we just spoke about with herd immunity, those that maybe even had the more mild smallpox infection still had an active infection happening in their body, and so then they would pass that along to other people. So by getting by having the variolation, then they were a person that was actively able to transfer this. And so then epidemics happened. And this is because it's so contagious. So that even if they had this really mild reaction and they had a mild version of smallpox, they still were able to pass it along because, again, they had smallpox in their body. But even though we don't do variolation today, we do have things called vaccines. And Edward Jenner, alive 1749 to 1823, is credited with developing our modern process of vaccination. So kind of taking this situation and turning it into what we now know as vaccination. This is because he observed that milkmaids who developed cowpox, which is similar to smallpox but more mild, were immune to smallpox. So he noticed that these women that were around cows that had this disease that is called cowpox, he noticed that then they could be around people that had smallpox and they didn't get smallpox. So they got cowpox earlier, but which was more mild and doesn't really have a high fatality rate. But then they could go and they could help people that had smallpox because they were immune. So then when he saw this, he saw the fact that these milkmaids were having cowpox but not smallpox. He hypothesized that exposure to a less virulent pathogen could provide immune protection against the more virulent pathogen. And this would be safer than variolation, because in variolation, remember, we're taking the fully functional smallpox pathogen and putting it into people on purpose. In this case, we're taking cowpox and we're saying this is much more mild and isn't causing this high mortality rate. We're taking cowpox and we can infect people with this, and then they're immune to smallpox, which would be great because smallpox does kill people. So in 1796, he infected a young boy with cowpox, and then after he recovered from this mild cowpox illness, he then purposely infected him with smallpox, but he did not get smallpox. So this was ethically okay in the day, not now, of course, but at the time, this is how it happened. He hypothesized this, he put it into practice using this young boy, gave him cowpox, then gave him smallpox, and he didn't get smallpox. So this is because it's genetically and antigenically related to the variola virus of smallpox. So our cowpox is very similar, and since it's so similar, our body is able to produce all of these memory cells in response to something that looks just like smallpox. And so this young boy didn't get smallpox. So since this was so successful, he was able to develop this smallpox vaccine. And so he took cowpox and he turned it into a vaccine, and he was able to then infect other people with cowpox, and then they were not getting smallpox. This meant that other people then started doing this. So other scientists were then working to develop vaccines. For example, Pasteur, he came up with vaccines for rabies, for cholera, and anthrax. And we're going to take a look at the different ways that we can make vaccines. But during the 20th and the 21st century, we ended up getting all of these different vaccines. So viral, we have chickenpox and shingles, which of course is the same thing. Hepatitis, measles, mumps, polio, and yellow fever. Our bacterial are diphtheria, pneumococcal, pneumonia, tetanus, and pertussis, or whooping cough. So during our 20th and 21st centuries, these are all of the different vaccines that were then developed because they saw how well this worked for Jenner, and then they were able to go through the process and weaken different pathogens and then make it so that we can make vaccines for them. So now, not all of the vaccines have this counterpart that is more mild, right? So the idea at first was that if we infect this person with cowpox, which is more mild but looks very similar to smallpox, that then they are not able to get smallpox. They're immune to smallpox. But we don't have, for all of these very dangerous pathogens, a more weaker version of the full illness, the full pathogen. So what we do is we go in and we modify them on purpose, and we modify them in different ways. So the first way that we're going to take a look at how we produce a vaccine, or the first class, I should say, the first class of vaccine that we're going to look at are called live attenuated. So a live attenuated vaccine is when we expose an individual to a weakened strain of the pathogen. So in this case, our goal is to establish a subclinical infection. So to establish an infection within the body, right? So something that the person has to fight off, an active infection, but it's subclinical. Remember, subclinical is where there aren't those signs and symptoms. So that will then activate the adaptive immune defense. So if we give somebody a more weak version of the pathogen, then the body can fight it off more easily. So we don't have those signs, we don't have those symptoms, but the body has fought off the actual pathogen. And since we fought off the actual pathogen, then we have the memory B cells and T cells and antibodies that are all within the body, so that if we then are exposed to it a second time, and we're exposed to, let's say, the actual full strength pathogen, that we would then be able to fight that off. So attenuation is decreasing the virulence of the pathogen. So that's why we're saying it's a live pathogen, but it's attenuated. It's been weakened. And there are different ways that we can weaken viruses. Or not viruses, I'm sorry, pathogens. One is through genetic manipulation. So what we can do is we can go in and we can eliminate key virulence factors. So we would go in genetically and make sure that we maintain the antigens that our body recognizes for that particular pathogen. But what we would do is go in and we knock out their virulence factors. Maybe not all of them, but some key ones that will then lead to the full signs and symptoms and perhaps death, depending on the type of pathogen we're talking about. So if we can knock out those virulence factors, then we can provide or produce that pathogen. We can make lots of copies of it and we can give that to people. And they'll have a full-on infection to it, but they don't have that virulence factor being made, and so they don't have those signs and symptoms and perhaps death. In this case, we have long-term culturing in an unnatural host or environment as another option for attenuation. So rather than actually going in and genetically removing pieces and making sure that we maintain the antigen piece, we can go in and we can just have these long-term culturing sessions. So if we take this and we put the pathogen inside of a culture, say a broth culture, and then we let it grow and grow and grow and grow, and then we transfer it to another one and let it grow and grow and grow. This is in its unnatural host. So meaning not within a body. So we're just growing it up in this broth, and it's going to be in an unnatural host or an unnatural environment. And then what's going to happen is it's going to promote mutations. So it'll mutate, and then we can decrease the virulence that way. So if we have mutations, because we're growing it up for so long and somewhere it's not super happy, those mutations can be mutations to their virulence factors, for example, because they might want to mutate or change their virulence factors, one, on accident, or two, in response to the fact that whatever they're doing isn't working, so they're going to mutate and change their virulence factors. And then this can decrease the virulence, because then when we put it in an actual person, it's not going to respond the same way that it used to. So some examples of our live attenuated vaccines are chickenpox are live vaccines. So they're live pathogens, but they've been attenuated. Same thing with German measles, regular measles, mumps, TB, typhoid fever, and yellow fever. So all of these are examples of our live attenuated vaccines. So when we have our live attenuated vaccines, this is one option, right? And we're going to look at all the different options. But when we use live attenuated, what we found is that it stimulates a more comprehensive immune response than our other types of vaccines. And this is because it is an actual live pathogen, and our body sees it that way. So if our body sees that we have this actual live pathogen in our bodies, it's going to mount a pretty large immune response to it. When it mounts this large immune response to it, then we have a more comprehensive back end, meaning that we have more memory B cells, we have more memory T cells, more antibodies that are floating around, so that if we get exposed to this pathogen again, naturally, that we have a better immune system response because our first response was such a strong response. In this case, we're activating both our cellular and humoral immunity because it is live. And so as a pathogen, it is going to try to get inside of our cells and reproduce inside of our cells. It'll be floating around in the humors and reproducing there. So it's going to activate both our cellular and humoral responses and therefore stimulate the development of our memory cells, so our long-lasting immunity. So the vaccination of one individual, however, can lead to natural transmission to other individuals. So this is why sometimes when a person gets a vaccination, they say not to be around people that are immunocompromised after the vaccine. This is because, again, similar to what we spoke about with herd immunity, if you're putting something that is live in your body, you're actually putting a live pathogen inside of a person's body, that person, when they then go out into the world, they are actively infected with that pathogen. They have that pathogen in their body, and it's reproducing, it's multiplying, because this is a first exposure. So we have an increase in the number of pathogen particles, whatever they are, virus, bacteria, etc., inside of the body. This means that that person can now pass that along to anybody they interact with. Now, of course, this may be a weakened version, or would be a weakened version, perhaps, unless it reverts back inside the body. But this would perhaps be a weakened version that they're passing along. However, this could cause a more active infection in another individual. So it can cause an active infection in another individual. It could be a subclinical infection in the other individual, and it could activate their immune defenses. However, if a person goes in to get a vaccine and they're, a healthy, youngish person that goes in to get the vaccine and say their body is doing just fine, fighting off this weakened version, if they go out and they're around people that perhaps do not have as strong of an immune system response or strong of an immune system in general. So if they're around somebody that is, for example, elderly or very young or just is already fighting something else off, so just a person that has, let's say, a regular healthy immune system, but right now they have a cold, if they are then interacting with somebody that has a vaccine, they could then get that particular pathogen in their body, and it could become very dangerous because if that person is already fighting something off, then their immune system is weakened. Or if they're just generally immunocompromised, their immune system is weakened. If they're elderly, their immune system is weakened. If they're young, perhaps their immune system is weakened. A live attenuated vaccine can then be passed on to other people because it is live and then can cause infection in other people. So this would be a disadvantage. This is on the next page. So a disadvantage of that, this is at the bottom here, is that there's risk of transmission during vaccinated individuals' subclinical infection to somebody that might be immunocompromised. So that's what we just talked about. So somebody that's unable to clear that infection. And again, immunocompromised doesn't just mean people that have some sort of disease that makes them immunocompromised, like HIV or lupus or Lyme disease or something like that. What we're talking about is somebody just in general that their immune system is not at their height. So elderly, pregnant, people that are fighting something else off, things like that. Some other disadvantages is that there are challenges that are associated with long-term storage and transport because they are live pathogens. So if they are live pathogens, it makes it really difficult to have them in storage for a really long term because as they are live pathogens, they need nutrition, they need to get rid of wastes, and then they start to die off. So it's difficult to keep these live attenuated vaccines in long-term storage, and that's important for transport. So if we're trying to get vaccines to other countries, that can be an issue. There is the potential for a patient to develop a serious infection rather than a milder one. So, for example, in somebody that's immunocompromised. And this may be somebody that feels like they're healthy and they can go into the clinic, wherever, and get the vaccine, but maybe they are already infected with something and they just don't know it yet. Maybe they're in day 1 or 2 of having some other infection, so their immune system is already decreased. Then they go in and they get a vaccine. They could have a more serious infection rather than the milder one that they're expecting. So then they would develop signs and symptoms of a disease during the active infection. So they could actually then feel sick. They could actually then get the illness that they're vaccinating for. And then the risk of the attenuated pathogen reverting back to full virulence. And I briefly said this a moment ago, where it's possible that once that attenuated pathogen gets inside of a person's body, that it's going to go along and do its multiplication thing. And as it does this, it's going to be fixing any mutations that might have happened. It's going to be gaining more virulence factors or developing its virulence factors. And so it could then revert back to its full virulence. And then that person would have the full virulence pathogen in them, likely then develop all those signs and symptoms of disease, be actually sick, and also be able to pass that along to other people. So at this point, then we're not concerned about if it becomes active. At this point, we're not concerned about whether or not it's a weakened one that we're passing along to other people. In this case, we'd be passing on the full virulence pathogen to other people, which even if somebody's not immunocompromised, perhaps then it would pass along to them. The next class of vaccines that we are going to talk about are inactivated vaccines. So inactivated vaccines are those that contain whole pathogens that have been killed. So in this case, we're not taking out the virulence factors and keeping in the antigen pieces. What we're doing is we're taking the full-on pathogen that is active, has all of its virulence factors, but then we kill that pathogen. Or we can inactivate it with heat, with chemicals, or radiation. So basically we go in and we kill or inactivate its ability to function. Now, inactivation can't affect the structure of those key antigens on the pathogen, though. So depending on the pathogen, we may have different ways of killing or inactivating a particular pathogen based on its key antigens that are on that pathogen. In this case, we don't have an active infection, so we get a weaker and less comprehensive response. So instead of the body saying, oh my goodness, we've got a pathogen in here, we need to go in and we need to do a full-on immune system response. All the B cells, all the T cells, all the antibodies, we need to fight this thing off and then have all those memory cells in response to that. In this case, we get a weaker response because in this case, we are putting it in the body and it's not going to get inside of our cells. So it only involves the humoral response because it's dead. It can't attach to anything and get itself into the cells in order to activate the cellular response. So it's only going to activate our B cells and our antibodies. So we get a less comprehensive response because it doesn't include those T cells. And it's also weaker because our body's not going to say like, oh my goodness, we have this full-on pathogen, we got to get it because it's multiplying and it's making lots of copies of itself. Our body says, hey, we've got this stuff in here. It's not supposed to be here. Let's just go clean it up and get rid of it. And so in this case, we have a much weaker response. The good thing is that it cannot be transmitted to other individuals because it's dead. So it's not multiplying, it's not going to those portals of exit, right? So it's not moving to the respiratory system where even if we cough because we have something caught in our throat, we're not going to pass that along to anybody because it's just going to be put into the bloodstream as the vaccine and it's going to stay there. It does usually require higher doses or multiple doses, however, because of this weakened response and this less comprehensive response. So this is when we would see something like booster shots. This is why we get multiple doses of something, is because our body will have this weak response, so then we need to kind of give it more of that pathogen to say, hey, look, we're seeing this pathogen again, we need to build up more of a response. See, we're seeing it again, we need to build up even more of a response. There are some risks here, not the risk of getting the entire full-on disease for whatever it is, full-on infection. However, it could cause an inflammatory reaction at the injection site because, our body is saying like, hey, there's something that's not self in here. We have that immediate immune system response saying like, let's go get it. But then, when the first cells are on the site, they're saying like, oh, we're good. It's just some stuff in here we got to clear out of the way. And so we have that response. It does cause our immune system to have that response, but it's not a full on like, these things are multiplying. We got to get a handle on it. So advantages of the inactivated vaccines is that long-term storage stability is there because it's already dead, so it doesn't matter how long it's going to be dead, it's still dead. And so it's easy to transport, so we can get these to other parts of the country or third world countries and things like that very easily. There isn't a risk of causing a severe infection because we know how much of the dead pathogen we're putting into the body. So there is not that risk of it multiplying and causing that severe infection. So some examples, cholera, hepatitis A, influenza, the plague, and rabies. So we have, these are different types of inactivated vaccines. The next type of vaccine, or the next class of vaccine, is called the subunit vaccine. So a subunit vaccine is when we expose the patient to key antigens of a pathogen. So in this case, we are not putting in whole cells. We're not putting in whole viruses. They're not live. They're not dead. What we're doing is we are producing just these key antigens. And we're doing this by chemically degrading a pathogen and then isolating its key antigen. So we want just those things that are going to activate our immune system. Then we are producing the antigens through genetic engineering as well. So we can go in, we can degrade the pathogen, we can isolate its key antigens, and we can use that, or we can just produce them through genetic engineering. We can go in and genetically make these pieces to then produce these antigens. In this case, the risk of side effects are very low, right? Because there isn't even a dead pathogen. We're literally just putting in these proteins, little tiny pieces, little proteins that are going to cause our immune system to fight against what it thinks is a pathogen that has these antigens on it. In this case, we don't have any chance of having an infection because there is nothing to do the infecting. It's just little tiny pieces. Some examples are anthrax, for these subunit vaccines, hepatitis B, influenza, meningitis, papillomavirus, pneumococcal pneumonia, and pertussis, or whooping cough. So the reason they're subunit vaccines is because what we're doing is we're taking these little tiny antigens, and likely, or oftentimes what we're doing is attaching them to larger pieces, so it's a subunit. We're attaching them to larger pieces that our body will then recognize, because remember, when they're too small, then we're talking about haptens, and haptens don't activate our adaptive immune response. We may have a more local inflammatory response to them, but it wouldn't activate our B cells or T cells. And in this case, we're not activating our T cells anyway because it's not getting into the cells. So again, it's going to be weaker, and it's going to be only activating our humoral response. If we look back here, some of these examples here, like pneumococcal and pneumonia and influenza. Influenza is a good one because we know that every year we have to get new vaccines made. They make new vaccines. People go out and they get new vaccines every year. And that's because we don't have the ability to have any antigenic variation in the vaccine itself. So some disadvantages, like inactivated vaccines, they have limited longevity. So sometimes multiple doses are required. Again, it's going to be only our humoral response because it's not something that can get into the cell. So we're only activating that piece, so it's not as comprehensive, and it's a weak response. However, we don't have any protection against antigenic variation. So we're just taking the pieces that look the way they do now and putting them in the body. Now, these pathogens could, over time, change the way they look. Remember, antigenic variation. And then, therefore, at that point, even if we had gotten the vaccine, it's not going to do anything for us. So, for example, so we can't account for that. There's no protection for that. So we are only able to take these little tiny pieces and say, now you are immune to this. But that doesn't mean if it changes in the future, we can do anything about it. So the next one is a toxoid vaccine. They contain toxoids, which are inactivated bacterial toxins. So again, in this case, we're not putting in the whole cell or the virus. It's not going to be alive or dead. In this case, this is used to prevent diseases in which bacterial toxins play a major role. So this isn't, of course, a vaccine that we do for lots of them. The examples down here are botulism, diphtheria, pertussis, and tetanus. So those that we've spoken about, those toxins. And that's because those toxins are what are going to cause these serious issues, these serious signs and symptoms. And so we use our toxoid vaccines for those to try to make sure that the body can have an immune system response against these very serious toxin-based diseases. In this case, we're activating humoral immunity, not cellular immunity. Again, we're not getting into the cell. And And also our humoral immunity is going to neutralize the toxins, right? Because we're not talking about the pathogen itself, we're just talking about the toxin that a pathogen produces. And so we activate our humoral immunity, our B cells are going to then produce antibodies that will attach to those toxins. Remember, we talked about the A, B subunit toxins. They'll attach to them and neutralize those toxins. And when that happens, then we'll have these plasma cells that are going to be producing more and more antibodies so that if we're ever exposed to that toxin again, then we can fight it off. Now, this doesn't prevent infection, though. So if we have a bacteria, say that responsible for botulinum, if we have that bacteria get in our body, it can actively reproduce and multiply in the body because we don't have immunity to that bacteria. It only sensitizes the immune system to the toxin. So we're just making sure that our body doesn't get overwhelmed with the toxin, but we can have an active infection with botulinum. The next one are our conjugate class of vaccines. Our conjugate vaccines consist of a protein conjugated to a capsule polysaccharide. So this is a type of subunit vaccine. This is developed to enhance subunit vaccines against pathogens that have protective polysaccharide capsules. So in this case, the antigenic piece of the bacteria is this polysaccharide capsule. So what they've been able to do is take this polysaccharide capsule and stick it to a protein so that we can enhance our immune system response to this particular pathogen. So remember that this is not as strong of an antigen, so our body may not recognize it as easily and try to get through it and get rid of it as easily. So this is important because these capsules are going to help them evade phagocytosis. So it's difficult for our bodies to clear an infection with those that have polysaccharide capsules. And so if we can make a vaccine that has that polysaccharide capsule and we give it to the people, then our body can be exposed to it, produce immunity, or create immunity to it, and then when we are exposed again, in that case, then we'll have an easier time fighting it off. It does cause invasive infections that can lead to various serious situations like meningitis or other serious conditions. Also, with these conjugate vaccines, children tend to respond better to these conjugate vaccines compared to our other subunit vaccines, which are typically ineffective for children. And the antibodies that are produced to both or there are antibodies that are produced, and they're produced to both the protein that it's conjugated to and to the capsule polysaccharide. So the children are able to respond better to it just because of their ability to respond to the polysaccharide capsule, which our bodies as adults aren't able to respond to as easily. And then they respond to both the protein that it's attached to and to the capsule because both are not self at this point. Disadvantages, they're costly to produce. It takes a lot of work to go in and create this conjugate, to create the protein to attach it to take the polysaccharide capsule, which when we're talking about polysaccharide, that's many sugars, that's easily dissolvable, so it's difficult to maintain that capsule and then get it to attach to a protein. There is no protection against antigenic variation, which we know from previous things. We can't account for any changes. It may interfere with other vaccines is a disadvantage. When it's given, it may cause some problems with some other vaccines. And then an example in this case would be meningitis from any of these pathogens here. So again, this is something that is important for those that have that polysaccharide capsule because it's really difficult for our immune system to overcome that polysaccharide capsule. So they try to make vaccines in this way to try to give us that primary exposure. So that is the last class of vaccine that we are going to talk about. So you should know all of the different classes of vaccines and, of course, the classifications of immunity or of our adaptive immunity, meaning the natural, the passive, the active, the artificial. And you should know the different classes of vaccines, their benefits, disadvantages, and what they are and how they work. And you should know some examples for them as well.